Okay. So I'll be speaking with you about revealing protein-protein interactions in large scale. So this presentation is based on the PhD work from Kara Johnson, who was a PhD student in our lab, and she defended her thesis and becomes a scientist here at La Jolla. And also this is joint work with co-first author Zhijie Qi, who is currently a bioinformatics PhD student. And also there is quite a few data points that I will discuss based on Irene Wen's work, who is a bioinformatics PhD student. She's rising up to her third year in her PhD. So the goal for this project is to discover as many protein-protein interactions as possible. So protein interactions are extremely important. Essentially, most of the functions of the protein, as we know, including signal transduction, forming protein complexes, all rely on some form of protein-protein interactions. Current methods for identifying protein-protein interactions remain extremely time-consuming, and to date, discover any protein-protein interaction would still require a significant amount of work. So there are at least two classes of current methods for detection of protein-protein interactions. So the one class, I would like to call them 1 versus 1, that would discuss a pair of proteins for their interaction at a time. This class would include those is2 hybrid methods, co-IP methods, taken the sub-network of some protein-protein interactions on the right-hand side. For example, a 1-to-1 method would test one edge between two nodes, one edge between two proteins at a time. For example, a co-IP experiment may test a potential interaction between, for example, the SERPINE1 protein and the THBS1 protein. So if we wanted to test all possible protein-protein interactions, it will take many, many parallel experiments. Another class of methods are 1-to-many. That would be able to identify all the interaction partners or nearly all the interaction partners for one center protein. This class of methods including pull-down mass spec and BioID and other methods. So these type of methods in one experiment would identify all the interactions that are centered at one protein. On the network figure on the right-hand side, if we were to take the SERPINE1 node, for example, so all the edges connected to SERPINE1 supposedly would be able to be reviewed by a 1-to-many method. So the bottleneck of the current methods is that it still takes a tremendous amount of time and funds to interrogate all possible protein pairs for possible interactions. For example, if we assume that the human genome contains 30,000 protein coding genes, if we wish to test for every pair of them whether there would be a protein-protein interaction, it would require on the order of 30,000 experiments for a 1-to-many method, or 30,000 square experiments for a 1-to-1 method to interrogate the entire space of all possible interactions. So the Kara Johnson's PhD thesis project is to develop a technology that is a many-to-many protein-protein interaction identification technology that hopefully would simultaneously review as many protein-protein interactions as possible in one experiment. So the central idea is to convert every protein-protein interaction into a unique DNA sequence, and then leverage the extremely high throughput nature of DNA sequencing to review the DNA sequences that represent protein-protein interactions, and thus decode protein-protein interactions by DNA sequencing. And so if I were to elaborate this idea, So the key is to label every protein with a piece of nucleic acids, such as DNA or RNA. In this case, shown here is a protein that is covalently linked to an RNA sequence. So if this can be done to many proteins at once, then we could imagine that possibly for all the interacting protein pairs, we might be able to connect their labeling RNA together to form a chimeric sequence, and then through RNA sequencing to simultaneously sequence the pair of RNAs ligated to the pair of interaction proteins. So that is the central idea of this PROPER-seq technique. So the PROPER-seq design is of following. So there are two major steps. The first major step is to take input cells and label as many proteins by a unique RNA sequence as possible. So hopefully all proteins are labeled by a unique RNA of that protein. And in the next step, we call it Inlet step, that is short for incubation, ligation, and sequencing. That would convert every pair of interacting proteins labeled pair of RNA into DNA sequences that are suitable for DNA sequencing. And through simultaneously sequencing this pair of RNA, we hope to reveal the original pair of interacting proteins. A paired end sequencing output. It is a sequence of two read pairs that are mapped to the genome. So every read pair is shown as a horizontal line, and its two ends are mapped to two positions in the genome. In this case, they happen to line up with the two genes in the genome. These two genes actually are on two different chromosomes. It's just that now that we have here approximately 40 to 50 read pairs that happen to be aligned to this pair of genes. So these 40 to 30 read pairs altogether would become the data or the evidence for us to say that the POP1 protein interact with the matrix 3 protein. So if I were to go over the entire process once again, so the PROPER-seq procedure would start from cells and harvest their messenger RNA pool. The harvest messenger RNA will be translated into a protein library with a modification, which I will specify in a minute. This modification would enable all the translated protein to be not detached from their original messenger RNA, so that the translated protein library would have the form that every protein are still conjugated with its own messenger RNA. Thus, the messenger RNA of every protein becomes this protein's unique RNA barcode. And in the next step, we call the Inlet step, we would harvest and stabilize and harvest all the interacting protein pairs, ligate their RNA pairs into a chimeric sequence of RNA1 and RNA2, and convert that chimeric RNA pair into a chimeric DNA sequence suitable for DNA sequencing. And so sequencing these chimeric DNA sequences, we have developed this toolbox to map them back to the genome and review protein-protein interactions and the entire protein-protein interaction network. So I will go into a little depth to the first step, which we call smart display. It is a display technology. in general, protein display technology is a technology to link a protein to a distinguishable nucleic acid component, such as the one shown on the right-hand side here. If we could successfully produce this protein RNA chimera, that would be the product of our display technology. The innovation from Kara is that she was actually able to make this display process one part experiment for all the genes in the genome, or as many genes in the genome at once, so that she will generate a displayed protein library in the following form. So how did she achieve this? The core of this idea is to leverage this toxin, pyrimycin, which if this pyrimycin is attached to messenger RNA, what happens is that after translation of forming the protein peptide, the translated protein peptide would be conjugated with the pyrimycin. If the pyrimycin is conjugated with the messenger RNA, which is our technique will achieve, then the translated protein will be conjugated with the messenger RNA. So by design, Kara designed this pyrimycin molecule that is linked to this specifically designed RNA sequence. And this RNA sequence actually is designed to be able to hybridize to a complementary RNA sequence that we would ligate to the 3' end of every messenger RNA. So now that consider we have a messenger RNA and we have a step to ligate this messenger RNA 3' end with this particular pre-designed sequence. And then we hybridize the other sequence. So the messenger RNA takes this particular form with now pyrimycin at its tail. So when ribosome starts to translate this messenger RNA, ribosome slides from 5' to 3' and start to make a peptide chain. So when it hits the end, the pyrimycin by nature will enter the A site of the ribosome and later will form a covalent link with the C terminus of the peptide chain. A ribosome will subsequently detach from this molecule and thus form a protein RNA conjugate. So this form is actually called RNA display or messenger RNA display. It is actually used for more than a decade. The innovation that Kara makes is to translate this protein display process into in a way that can be systematically display as many genes as possible. So in Kara's setting, in the smart display process, her input is the entire messenger RNA library retrieved from input cells. The following steps are going to convert the messenger RNA into DNA by reverse transcription, but also insert certain sequences at the 5' and 3' ends of the DNA. This process is the same experiment in the same solution of every messenger RNA. So she doesn't have to deal with one gene at a time. She deals with all the transcriptome, entire transcriptome at once. So for every gene, what Kara achieved is to add some translation required sequences to the 5' end and some sequences that are required for the pyrimycin sequence to be hybridized onto, and some flag sequence to select for the translated protein. So now with this new library that is made from the input cell's messenger RNA library, she can translate and then form a library of these displayed proteins. Each now is conjugated with its own messenger RNA. So the next step from here is the inlet step. So what the inlet step intends to do is to convert every pair of interacting proteins into a pair of ligated DNA sequence suitable for paired end sequencing. So what she does in that is that she now has a library of displayed proteins. She stabilized that library into streptavidin beads just for later steps of washing easier for her. And she reverse transcribed RNA into double-stranded DNA. So now every protein is now labeled by the cDNA of its own messenger RNA. Now she incubate this library with another library made in the same form. So this is a second displayed protein library of many proteins. And in the second protein library, after incubation with the first protein library, supposedly some protein interactions will happen and some protein complexes may be formed. She stabilized the protein interactions by cross-linking amino acids only, wash the uninteracting proteins and nucleic acids and whatever in the solution. And then she would have resulted in some cross-linked protein interactions stabilized on beads, each labeled with cDNA. Then she ligate the 2 cDNA sequences per protein pair into this chimeric DNA1, DNA2 form, and she harvests those DNA and convert them to sequencing library. And after she sequenced those chimeric sequences by paired end sequencing, we Zhijie designed the PROPERseq tools to conduct quality control metrics and statistics to pull interactions from these chimeric sequences. In a nutshell, the PROPERseq tools would take the input read pairs and train those artificially incorporated sequences into 2 ends, do quality control, and map each end to the genome separately. They typically do not map to the same part of the genome. They can map to different chromosomes, and deduplicate and remove duplicate sequence, and then identify gene pairs that are supported by a sufficiently large number of read pairs through a statistical test. So this would be association test. And then identify the protein-protein interactions. So this code base is deposited at the following DOI. And so here is an actual experiment that Kara conducted. She did 6 experiments on 3 cell types. So the 3 cell types are HEC, these are embryonic kidney, JERCAT, those are T cells, HUVEC, those are endothelial cells. This is the most inside layer of our blood vasculature. And so she did 2 experiments per cell type, and she had 6 experiments altogether. And each experiment yielded approximately 200 to 400 million read pairs. This would be the typical Illumina sequencing on 6 lanes of a flow cell. And using this data through PROPERseq tools, we reviewed 200,000 protein-protein interaction pairs. Those involved more than 8,000 proteins. So the right-hand side is Irene's graphical representation of the entire collection of 200,000 PPIs of the human genome resulted from the 3 cell types. So all the database documented PPIs of the human genome to date, there are approximately 150 PPIs documented through all high throughput, low throughput curated experiments. And so these experiments contribute approximately the same number of PPIs to all previous documented PPIs to date. So if we were to zoom in to see some specific part of the protein-protein interaction networks, so here are 2 parts that Irene pulled out. One is the subnetwork of the PPIs related to translation. So these translation-related proteins, every edge represents a PROPERseq reviewed interaction. It turned out that those proteins can be roughly grouped into 4. One group are the ribosome small unit, the other ribosome large unit, subunit. Those are very much expected to be there and interacting. And the pink edges are the interactions that were identified previously. through either high throughput or low throughput or biochemical approaches. And there are translation initiation factors. There are also translation elongation factors that now form a subnetwork of interactions. So on the right-hand side is a subnetwork of protein interactions involved in RNA splicing. And that is a very important machinery that traditionally was known to involve, for example, the splicing complex ABC, the uSNRPs, those are the uRNA binding proteins, heterogeneous RNA modification particles. Those are known to some extent involved in RNA splicing. Nevertheless, the entire process and machinery is heavily under pursuit. Now that here we also see how those SM proteins, SR proteins that are involved in co-transcriptional and co-splicing modification of the RNA are also involved in the process. So if we go back and look at the 3 cell types, we would recognize that although we put all the data from the 3 cell types together, but after decoding protein-protein interactions, we can still ask the question of where did the read pairs come from which experiment? If the read pairs came from all the 3 cell types, we may infer that this protein interaction could have happened in all 3 cell types. But if the read pairs came from only 1 cell type, for example, or primarily from 1 cell type, it basically means that that gene pair was only expressed in that cell type, at least in the 3 cell types. So that type of information could represent cell type specific protein interactions. So here we simply dissected these 200,000 PPIs into the following categories. They are either shared or tend to be more enriched in one of the 3 cell types. So you can see that approximately 60% to 70% of the PPIs perhaps are shared, but there may be somewhere between a quarter to maybe 30% of the PPIs perhaps can be attributed to a particular cell type. If we were to plot the subnetworks into this 3-dimensional plot, how to read this plot is that every dot is a gene ontology term. And these gene ontology terms position would be coded in the following way. If the PPI is identified by PROPER-seq, the amount of the proteins that are identified by PROPER-seq belonging to that gene ontology term would decide where this dot is in this 3-dimensional figure. If all the read pairs contributing to the proteins in that gene ontology term were from HuVec, then this dot will be dragged toward the HuVec direction. The more proportion of reads supporting the proteins of that gene ontology term in HEC, that dot's position will drive to the HEC direction, and so on for the JERCAT direction. So the relative position of that gene ontology term represent the relative support of read pairs from PROPER-seq from the 3 cell types. If there is about the same support from all 3 cell types, that gene ontology term will be somewhere in the middle. What we see here is that most of the terms are somewhere in the middle, suggests and coincide with the impression that most of the read pairs were generated from all 3 cell types. Nevertheless, there are some terms that seem to be strongly supported by HuVec, but not by the other 2, or there is like 1 or 2 that are strongly supported by JERCAT, not supported by the other 2. If we were to take about the 2 of the JERCAT specific gene ontology terms, those 2 terms are positive regulation of T cell proliferation and T cell activation. JERCAT is a T cell line, So that it perhaps is no surprise that the PPIs or the read pairs supporting PPIs are associated with the gene ontology term related to T cell function. And those are the reviewed PPIs specific, more specific to JERCAT. You see that most of the PPIs are JERCAT only a few in gray that are shared by other two cell types. The edges in pink are JERCAT specific interactions, having a lot to do with the high mobility group of transcription factor that very recently has been implicated to a lot of the T cell activation. And in the HUVEC, the endothelial cells of the blood vasculature is very critical for regulation of many things. As you may know that, for example, the COVID virus actually harm the vasculature from the inside. And many of the problems, like, for example, Alzheimer's would have something to do with a blood-brain barrier, or the barrier by itself is formed by the endothelial layer. So there turn out that the goal terms that are specifically reviewed by the HUVEC read pairs are involved in extracellular matrix, assembly, disassembly, and cell mobility. These are the perhaps most important intrinsic natures of the most inside inner layer of the vasculature system. That is the endothelial layer formed by the endothelial cells. There are reviewed endothelial cell specific functions, including those are associated with THBS1 protein, SERPINE1 protein, and their involved interactions. These now retrospectively actually perhaps tell us a lot about the pro-inflammatory response of the endothelial system that perhaps is one of the most important response of our blood vasculature. One last thing that Irene contributed that perhaps is interesting is to ask the question: is there any correlation between genetic interaction and a protein-protein interaction? Protein-protein interaction is a kind of physical interaction between two proteins, whereas genetic interaction is an inferred type of interaction through genetic evidence. For example, the synthetic lethal gene pairs is a type of genetic interaction. If two genes, one deleted at the same time, it confers a lethal phenotype to the cell. So those gene pairs are called synthetic lethal gene pairs. You see the definition is by genetics, right? So there is a heavy pursuit of trying to get what are the synthetic lethal gene pairs in humans. Apparently, this may not be just straightforward from experimental studies, but there are recent developments of using both experiments and bioinformatic methods to inform us of the human synthetic gene pairs. So Irene took overlap between the inferred human synthetic lethal gene pairs and the human protein interactions identified by the method, by PROPER-seq, and these are the intersection. So what Irene finds is that in the intersection, those are both PPI and genetic interaction synthetic lethal genes. So those synthetic lethal gene pairs tend to be more heavily impact at the hub proteins in the identified human protein-protein interaction network as compared to the entire network. And so if I were to summarize and thank Michelle for the notice, so the PROPER-seq maps protein interactions in large scale. And it actually contributes to more than 20,000 novel PPIs to the community. We experimentally tested a subset of them, which I didn't have time to go over each. They are involving methods for inferring interacting protein pairs from the human genome based on bioinformatic methods, including protein structures. Many of those computationally predicted PPIs didn't have experimental support. Here, we contribute experimental support to 17,000 of those computationally predicted protein interactions. And also we observe that the synthetic lethal gene pairs tend to more impact on the hubs of the human protein interaction network. So the work just appeared last week in Molecular Cell online. Hasn't been in print yet, but it's available through the Molecular Cells website. So I mentioned that the core group from Kara, Zhijie, and Xinzhao Irene, and as well as I would like to acknowledge the other authors in the team. With that, I would like to thank everyone. I do not know if there's time for questions. If not, I'm happy to take it offline.
Thank you so much for your talk, Professor Sheng. I actually saw the paper come out last week and it was really exciting to read.